slightly different, but walking hand in hand with degenerative medicine. Okay, what are telomeres? Repetitive sequences. The only thing that I want to say about this is, if you notice, this is actually one of the strands. There's two complementary strands for most of the telomere. There is a short segment that is uncapped, backward, that is unmatched at the end of the telomere that's folded back on itself, so that it is not uncapped, it's protected. In this sequence of TTA-GGG, there are three guanine molecules, and guanine in particular is extremely sensitive to oxidation. So by nature, the telomere is extremely sensitive to oxidation, probably more so than most segments of other areas in DNA. So you have 92 telomeres. I looked at this slide. This is actually mostly no past presentation. I looked at this slide and I wasn't sure. So I guess this is, let's go to the Russian world heavyweight boxing. We're going to focus in on him, and here's the skin. Here are the many skin cells, many nuclei. This looks familiar as it's part of your presentation. Lots and lots of cells. People argue about how many. Some people say 9 billion, some people say 9 trillion, some people have other numbers, depending on whether you count the hematopoietic component, including the circulating cells. As you focus in, you go into the nucleus. These are the chromosomes. And then as you focus on the chromosome, you see it's got 4 little blue end tips. Those are the telomeres. We're going to highlight them in yellow. And we're going to do something called an HT glowfish. This is also the lifeline assay. This is an actual picture of fluorescent chromosomes using that technology. And the fluorescent aspect, the highly fluorescent aspect, is the telomere end. So you can see the difference in how much telomere DNA you live on versus how much genetic information you actually have. This is the HT glowfish picture. What do? Most of you know this already. They serve to protect the integrity of our genes. I think that's very, very important, because it's not just at the end of the chromosome and what we call the subtelomeric region that's associated with it. It's the whole genome, end to end, can be affected negatively by a shortening of the telomeres. And you may have a mutation in it, smack dab in the middle of the DNA strand because of a short telomere problem. It's possible. They keep it from degrading to prevent fusion, which is what you see in cancer. You see shortened chromosomes. You see what's called the ALT lengthening mechanism, where one chunk of a chromosome attaches to another. You see dicentric fusions and donuts and all kinds of ugly looking things that are either the precursors or in some cases, the hallmarks of cancer. They allow cells to replicate because as a biologic time clock, when your telomeres get too short, there are two checkpoints, if you will, that should, when they're functioning properly, move the cells from the replicative cycle into either senescence and apoptosis, one or the other, depending on what cell it is, because some cells preferentially apoptosis, some cells preferentially senescence. It's usually shown as a line, and somewhere around 5,000 base pairs you're supposed to get into senescence, and somewhere around 3,000 you're supposed to apoptose, but that's not specific. That's a sort of a simplification of what's really happening. It varies from cell line to cell line. All right, bottom line, they protect cells from mutations, senescence, and death. Now, it's important for you to understand that mutations can still occur in the DNA, independent of telomere shortening. But this has been associated with many of the diseases of mutation that we see that develop through our lives, as opposed to those that we inherit. It determines cellular age. People would argue with this a little bit, but I don't. Somatic cells do not have, as a whole, a lot of telomerase or any telomerase expression. So if you think of telomerase as a gene for immortality, the reason I say that is because we have created immortal cell lines by turning on telomerase, even though we haven't created immortal nowadays as humans yet. The somatic cell is repressed, it's turned off, with the exception of highly replicative tissues, which include the hematopoietic components, blood, white blood cells specifically, the liver, intestine, the skin, and some other compartments. Some areas of the body have very little replication, as far as we know. Those would include the brain and heart, so-called post-mitotic tissues. However, telomere dysfunction and telomere shortening can directly affect the health of those cells as well. That's been shown in the past few years. So most of our cells are subject to this mortality, this biologic time clock. They have little no telomerase expression. Telomere length shortens with each replication. So this is something you really can't escape by exercise and diet and meditation and all those other things. So I want to clarify something for some of you today. Two things. Number one, arresting or slowing down the loss of telomere length is very, very, very different than turning on telomerase and lengthening the telomere, or adding any length back to the telomere, even if it's not enough to compensate for replicative losses. Two very different things. And you should do both of those things, and that's what our book's about, and we'll get into some of that more today. We're conceived with 15,000 base pairs. You know that from Bill's lecture yesterday. And you also know that that essentially means that the clock starts when we're conceived. We're not dying from the moment we're born. We're dying from the moment we're conceived. By birth, because of extremely rapid replication and the growth from the zygote, term I learned about 35 years ago, into the actual embryo, there are billions probably of replications that make that happen. And we use a lot of telomere length up there. We use 1/3 of our telomere length up. But in point of fact, since cells generally die at 5,000 base pairs, we actually only have 10,000 base pairs to live on. And we use 5,000 of those up just getting from the zygote stage to the newborn. And there's your answer as far as what happens to the rest of them. And that triggers one of those cellular fates, cellular senescence or apoptosis. So if we think of the cell as a miniature representation of us, our cellular health is extremely important, and what determines our cellular health determines to a large extent our health, if enough cells are affected. So telomeres are considered the biologic clock. Telomere shortening regulates how many times a cell can replicate itself if the cellular checkpoint mechanisms are in place. If they escape the cellular checkpoint mechanisms, you basically create a cell that is headed or has become cancerous. Every time the cell replicates. So accelerated replication is one of the things that may shorten telomere loss. And what causes accelerated replication? Accelerated damage causes that. So the more damage you do to your cells, the more you're asking them to repair. And I think most of you know many of the large determinants of cellular aging and personal aging. Include things like sleep, diet, exercise, stress, and toxin and environmental exposures. You know this from what I just said. When telomeres get too short, their fate is simply to turn on either cellular senescent mechanisms or apoptotic mechanisms. If they escape, you're going to run into serious, dangerous problems. And again, telomere length represents your biologic cellular age instead of your biologic age. This is just a representation of what actually happens. We start out with a certain amount, we lose that, and then bad things begin to happen. Now, there's something that we're going to talk about a little bit as we see the shoelace untie and the aglet, if you will, erode. And that is the concept of senescence. And this is something that's extremely important, I think. And you know, the result of senescence and cell death is that cells age, and we lose potentially cellular mass if we're not able to replicate enough. And so we begin to see organ dysfunctions down the road from the beginnings of these problems, as the repair processes are no longer able to keep up with the damage process. It's thought that the damage process is happening at about the same rate throughout our lives. It must be accelerated by mitigating those things: sleep, stress, diet, physical exercise. But I think personally that the cellular senescence is actually one of the things that's driving aging, because Bill and I have a mutual friend by the name of Michael West, who owns a company called Biotime, and they soon again own Geron. And Mike was also the founder of Geron. And Mike in 1996 published a paper on something he called sense-static activation. He was one of the earliest people to document that when a cell goes into senescence, it's not just sitting in the rocking chair, removing itself from the cell cycle and waiting to die. It's actually expressing inflammatory cytokines. And those inflammatory cytokines are not just limited to intracellular or the immediate cellular mobility. They can spread out like a pebble of water